sixth year biology students, there is a chapter on your Leaving Cert course for June 2026 titled DNA and RNA. And a lot of people get stressed about genetics, so in order to understand genetics, you first need to understand DNA. So we're going to talk in this video everything that we need to know about the structure and function and what it looks like and what the basic units are called. So first of all, the word DNA. DNA stands for deoxyribonucleic acid. DNA is always found in the nucleus of a eukaryotic cell because remember, eukaryotes have a membrane-enclosed nucleus and membrane-enclosed organelles. Prokaryotes, like a bacterial cell, don't. So DNA will be found in a DNA loop as opposed to in the nucleus. So DNA is always found in the nucleus of a eukaryotic cell. Now, there are some other non-nuclear locations of DNA. For example, the mitochondria and the chloroplast contain their own DNA. But DNA, if we look at the structure of it, is a double-stranded, what's called a helix structure. So here we have the 3D structure of DNA. Now this DNA molecule is too large to fit out through the nuclear pores, which is what you're going to cover in protein synthesis. But for this video, if we focus on looking at one of these two strands of DNA and then untwisting it and looking at a 2D version instead of a 3D version, you will see that DNA is a long section with all of these little things that are jutting out to the side, which are known as bases. So the basic unit of a DNA is a polymer, and a polymer is made of a basic unit known as a monomer. And the basic unit of DNA is referred to as a nucleotide. The nucleotide of DNA is made up of 3 sections, which in past exam papers you've been asked to label and draw. So it is made up of a phosphate joined to a 5-carbon sugar called deoxyribose. Then it's also joined to a nitrogenous base. There are 4 nitrogenous bases that you can have in DNA: adenine, thymine, guanine and cytosine. Now these bases are grouped. We have purines and we have pyrimidines. Purines, adenine and guanine, pyrimidines, thymine and cytosine. And I always remember the pyrimidines have a Y, thymine and cytosine have a Y. Now, if we look at one basic monomer known as a nucleotide of DNA, they're going to combine to another nucleotide and another one to make up the polymer. So how do they do that? The phosphate of one nucleotide bonds to the sugar of the next nucleotide. So it creates a backbone made up of sugars and phosphates. And that's the backbone of the DNA helix that we see, a sugar-phosphate backbone, because the phosphate of one nucleotide combines and bonds to the sugar of another. Then we have all of the bases jutting out, which I talked you through a second ago. But when these bases, because we have 2 strands, and it's the bases that are going to be where the 2 strands kind of pair up, if you will, and they'll bond together with what's called a hydrogen bond. But only certain bases can combine together, which is called complementary base pairs. So adenine can only bond to thymine in DNA, and guanine can only bond to cytosine in DNA. So they are our complementary base pairs. If you were given a DNA sequence and you were asked to write the complementary sequence, that is something that you need to be able to do in your exam. So if we do an example here, we're going to have C, A, T, T, C, G, T, G, A. If that's the sequence we were given and asked to find the complementary base pairs of this DNA sequence, our answer would be G, T, A, G, C, A, C, T. And that would be a complementary base pair of this DNA sequence. So if we look at our DNA sequence for a minute, when we're talking about our bases, in our protein synthesis and in our DNA chapter, we group our bases into groups of 3, which gives us a codon. Now, what are they coding for? Every group of 3 bases is coding for a particular amino acid. And eventually, those amino acids, which you'll see in protein synthesis, will come together in a chain and fold to form a protein. But because we have unique sequences of codons, that means we're making unique proteins. So every time we get our codons together, we're not necessarily making the same sequence of amino acid or the same protein. So bear in mind that the different proteins that we can have would be the likes of enzymes, which control metabolic reactions, hormones, which you do in the endocrine system chapter. You can also have structural proteins like keratin or myosin or collagen. So just bear in mind that you're not making the same amino acid each time, because you have unique groups of codons that will code for particular amino acids. And depending on how those amino acids join together, that will determine after folding what type of protein you're going to have and what job or what role that protein is going to do. So that is DNA, deoxyribonucleic acid. It is found always in the nucleus, and some other non-nuclear locations are the chloroplast and the mitochondria. So if you're studying Unit 2, make sure you understand this before progressing on to RNA, which will be next.